Good day, everyone. I am Eliseo Pangilinan, and today I am presenting to you my chosen topic for Biology 101, and it is about polymerase chain reaction. I will be discussing a brief introduction to it and its application in modern medicine. My main objective for this topic is, as I mentioned earlier, to discuss the application of polymerase chain reaction in modern medicine. Other objectives are to discuss the history of PCR, brief overview of PCR processes, give an example of PCR application in clinical laboratory, and share my own conclusion at the end of the discussion. Let's begin by tracing the history of PCR. The isolation of top DNA polymerase from Terbus aquaticus marked the start of modern PCR. This is a significant breakthrough for molecular biologists as the Taq polymerase is a thermostable enzyme capable of repeat PCR cycling. Before I dig deeper into our discussion, let me give you a definition of PCR by the National Human Genome Research Institute. PCR, an acronym for polymerase chain reaction, is a laboratory technique for rapidly producing or amplifying millions to billions of copies of a specific segment of DNA, which can then be studied in greater details. Another term for PCR is DNA amplification because it involves primers, which are synthetic DNA fragments, to select a segment of the genome to be amplified, and the repeatable cycling process to amplify the segment. Now that we know the meaning of PCR, let's dig deeper into the elements needed for PCR and the sequences of how PCR works. For PCR to start, polymerase is needed, primers, DNA template, nucleotides, mix buffer, and PCR tube. In the image shown, you see a machine and it's called a thermal cycler. With the proper things included to start the PCR process, PCR sequences start first with denaturation, next is annealing, and then extension. That is the overall process of one cycle. Repeated cycles produce millions of copies of DNA. As I mentioned earlier, denaturation, the first step in the PCR process, involves the Taq DNA polymerase and heating up to 94 to 96 degrees Celsius for 10 to 20 minutes. The heating causes the two complementary strands to separate, thereby being tagged as a melting step. Next step is annealing. Here, the temperature is lowered to 45 to 60 degrees Celsius. The temperature is critical and needed for the primers to bind to the complementary sequence on each of the DNA strands. These primers are designed to attach to the target of interest. In this step, the region of sequence that now lies between the two strands is called amplicon. This is the last step in the PCR process, in which called extension. In this step, the DNA polymerase peels the missing strand. The temperature applied here is 72 degrees Celsius and is dependent on the stability of top polymerase. The whole process takes about 45 minutes and repeated cycling would generate millions to billions of copies. Now that we all know how PCR works, let's go to the main topic. The most common application of PCR techniques in modern medicine: quantitative PCR, qualitative, conventional, standard, and multiplex. Among these, quantitative is the most common, especially used in laboratory techniques. Giving an example, it was helpful during the COVID-19 pandemic. Another term called for its real-time PCR because it gives a result in a matter of time. In this slide, I enumerated the uses of PCR: cloning of genomic DNA or cDNA, mutagenesis or modification of DNA, assays for the presence of pathogens, detection of mutation, analysis of allelic sequence variation, genetic fingerprinting or forensic samples, nucleotide sequencing, Here is a photo example of a machine using PCR in laboratory setting. This machine is called Gene Expert. The technique used here is RT-PCR. This is used for testing strep A, COVID, RSV, flu A, flu B, gonorrhea, chlamydia, and group B strep. The required samples must be submitted acceptable for the specific test. Clinical laboratory department in hospital find this a helpful tool to aid in patient care management. Here is an example of multiplex PCR. It tests COVID, flu A, flu B, RSV all at once in one cartridge. In this video, it shows an example of strep A cartridge being pulled out of Gene Expert after processing. This is a single testing RT-PCR use. So this video is an example of a single testing of COVID PCR. The sample required is a nasopharyngeal swab and testing takes about 35 minutes. Here's my own conclusion. With all the principles of PCR combined and proper techniques, this is a significant milestone for modern medicine and molecular biology. In all technology, there's an advantage, limitation, and disadvantage. But with proper tools and right application of science comes bigger breakthrough for polymerase chain reaction. This is a useful tool for everyone in healthcare field and patients as well. That wraps up my topic presentation. Thank you for watching.